Okay, so today we have lecture number 7. So we got past the modules about DNA sequencing and data processing. So now we are going to assume that we will be able to process and analyze kind of sequencing data, and we want to look at more complex kind of research situation and more analysis model. So today we're going to start with the phylogenetics, or the study of evolution. So today's agenda is simple. We're going to talk about why evolution, like why do we talk about evolution, and the techniques that we need and the models that we want to understand to reconstruct the phylogenetic tree or the evolutionary history. So the reason why we want to talk about evolutionary perspective here is because nature is the largest kind of laboratory experiment that you can find, right? So from monkey to us, like, we don't have to try to mutate the monkey to derive the human. Then we already have that process through nature, and we can study the difference in DNA, in gene expression, in any molecular kind of aspects to understand the difference between us and kind of other species. And the key idea is kind of mutation occurs all the time. They occur randomly. They occur without knowing what the consequences will be. But through the process of evolution, mutations that are beneficial remains to be observed. Mutations that cause disease or not beneficial kind of disappear over time. So that's why I say kind of nature is a largest experiment that you can study. So that means that you can get a lot of knowledge about biology, knowledge about protein function by comparing genomes and DNA across species. So before we go into the molecular details, let's think about the ideas of evolution. So we have kind of mutation that occur randomly on the genome, like DNA damage happens all the time. But sometimes that change in DNA happens to cause change in phenotype that fits the environment, that fits the habitat, that fits the situation for that organism. So birds kind of that live in different islands with different food, like different tree, different fruits, they develop different beak, different mouth basically. So that's kind of controlled by some genes, right? And when birds have mutation that cause their beaks to change, if the change happen to match the type of food that they need to eat, right, that allow them to utilize more resources. And that allow the birds that are more successful, like the birds with the mutation, if they became more successful than their peers, then they produce more offspring, and then their offspring now carry that same genetic mutation. So the process of natural selection has two steps. Mutation occurs, and then mutation that are beneficial got selected through the process of nature. So this means that if we take genomes or DNA from different species and we compare them together, we can identify genes that are responsible for certain phenotype or for certain disease. Even before having to do any kind of wet lab experiment, we can compare, for example, like when we study genetic disease, we are comparing DNA from among humans between the one with the disease and the one without. So we can tell that any genetic mutation that occur in normal people, like healthy people, probably will not contribute to the disease. But any mutation that we found consistently, like we found many times in patients, like those will be linked to the disease. So that's the concept that we can compare genomics information, and we understand the function of the gene without having to do any experiment. We get some candidates just by comparing the genome. And if you want to study brain development, like people use primates, like monkeys, as the model because they are very close to us in terms of the evolution, but the brain developments are different. So genes that help humans develop our brain, like, has to be something that are present in human, but not present in most monkeys because they don't have the same development. And if you identify like some enzymes like in bacteria or in plants, and you want to know which enzymes is responsible for that activity, like you want to extract it, you want to modify it for any kind of biotechnology purposes, you have to compare the genomes of the bacteria or the plant that have that enzyme against the plant and bacteria that do not have the enzyme. And then you can identify like which gene got maybe duplicated or maybe which gene got integrated into the microorganisms or the plants that have the no enzymes. So that kind of gives you information about the candidate gene, and maybe sometimes it even gives you ideas of how to edit the protein. Like if you compare the enzymes, like if it's a family of enzymes, you compare the protein and the DNA sequence across different organisms that have the same enzyme, right? And you are asking, if I want to modify the activity of the enzyme, which amino acid position should I mutate? You can compare if there is a position that's conserved, like over time in any organism, it's always alanine. Right? If you mutate that position, it might disrupt the entire protein. So that might be the position that you don't want to try. But if there is a position that kind of change in some organisms, but not in the others, then that might mean that it can change the specificity for the enzyme to be for different kind of habitats or different environments. So that might be position that you want to introduce more mutation and do laboratory screening to see if you get a new activity or not. So comparative analysis using kind of evolutionary perspective can be an important tool for you in any molecular study. So to give you some example, some inspiration. So this is a study where the student have a very specific interest. So he came to me and he said that he want to understand how placenta develop in higher order mammal, like because for species like birds, like they have eggs, like for crocodile they have eggs. Like, these don't have placenta, but in mammal, like we have this kind of organ. So the students want to know which gene kind of contribute to the development of new organ, right? Because this is not like a subtle a new gene, like new molecular function. It leads to an entirely new ways of reproduction with new organs. So the idea, like if you are a classical molecular biologist, like you might try to develop a mouse model, like mouse have placenta. You might try to introduce mutation to the mouse, like, and hopefully find a certain strain that have defect in their placenta. And then you try to identify which mutation lead to that effect. When we think about that, it's possible, like, but we feel sorry for the mouse. So another way to narrow down on which gene that we should study in the context of placenta development is if we have genomes from many species, from many animals, many birds, many crocodiles, many species, like we can compare their genomes. And species without placenta like, will serve as our control. Like, genes in this species probably are not going to be essential for placenta development, most of them. And genes that are unique to mammals with placenta, like these are genes that can be candidate for the placenta development. But we also have to think that it's not only placenta that develop in later mammal, like we also have other phenotypes, we have other molecular pathways. Yeah. So there's going to be thousands of genes that are unique to us and not in crocodile, not in chicken. But we have another clue. Like, another clue is that genes that are important for placenta development has to be acquired or has to be emerged at the timing when we separate from crocodile, for example. It shouldn't be a new kind of recently acquired gene. It should be genes that were acquired by the time that we have the common ancestor with crocodiles. So we have time concern that any gene that we are interested in, the time that it enters our genome should trace back to that long in the past where we feel kind of friend with dinosaurs, basically. So the student kind of takes the genomes for all the animals that we can find, that we know nowadays, like There are 267 of them. And we reconstruct the evolutionary history, kind of which species diverge from which species at which time point, like we can reconstruct. That's the technique that we are going to talk about here in today's lecture. But let's say if we can do that, right, we can trace back the evolution to the time where we are still the same as a crocodile. And the student can model the development of placenta in two steps. First, like the genome doesn't have the gene, like we don't have the gene for placenta development. But at a certain point, like there might be some mutation, there might be some integration from the viral sequence, there might be some duplication in the genome that create the gene, the new gene, that will be responsible for placenta development. But when the genome first acquires the gene, like, it might not be properly controlled. The gene expression might not be properly regulated. So if we have, in addition to just the genome sequence, if we have the gene expression data, then we can also identify which gene emerged in our genome back in the past and also show the expression. Right? It's not just a silent gene. It's a gene that became activated, gene that became expressed. So the student also collect RNA sequencing data for this species and then analyze kind of which gene combined with the DNA sequence. Right? So we know which gene emerges after we diverge from crocodile. And we know which gene became expressed, like became active after that, right after that. So through this process, the student identify maybe 3 or 4 genes, if I remember correctly. And some of them, people who do wet lab experiment can interfere with this gene in mouse and then can show that if we knock down the gene or we knock out the gene, the mouse will have a dysfunctional placenta. So we can identify kind of candidate genes where we can do experiment to confirm without having to screen a large number of mutations. Right? So we kind of save a lot of mice by doing a good computational analysis beforehand. Another example, which if you might have seen this before, but if not, it's something that very exciting for in terms of the evolution of human. So if you compare the human to other animals, like other animals, like usually in the sea animal, they should usually have smaller number of chromosomes than us because they are not as complicated. Mammals and human, we are kind of more complex. Like we need more genes in order to have all the function that we need. Like so, in the past, like at the start of the evolution, there's not going to be a large genome. So at a certain point in time, species at that time only have, let's say, 17 chromosomes. The only ways that they can become more complex kind of species with larger genome is that the genome has to expand somehow. And people want to understand it, so they compare kind of our genome to the genome of other sea animals and other kind of less complex organisms. And they deduce that there was 2 points in time that the genomes duplicated, like from 17 became 34. And from 34 became close to 60. But today, like we lost some of those chromosomes, so we can revert back to about 23. But in the past, there was a period where evolution kind of exploded. It's like a big bang of evolution. So the genome content kind of doubled twice. And that kind of quadruple in number of genes kind of allow the adaptation of new gene function that became kind of what we have kind of today. So that because we cannot use time machine like to go back to the past, the only way that we can infer such kind of event that happened long ago, like millions of years ago in the past, is through computational modeling. And this is some of the techniques that we are going to talk about today. A more recent example, like the COVID-19 that we just had 5 years ago, like when people start to collect more DNA and protein sequence from coronavirus, people start to wonder what will the next strain be? We have alpha, beta, gamma, delta, omicron. Right? We know that the virus is changing. And we know that every time it changed, it got worse for us. So people want to know like, what happened. Can we tell if which direction will the virus evolve in the future? How does it escape our drug? Right? How does it escape our vaccine? So we can do this with evolutionary analysis. Right? We can look at the protein and DNA sequence of coronavirus like from day one from Wuhan compared to the new sequence that we collect all over the world. So we can see the position of the protein that change over time. And because we know the timing of the sample, right, we can compare the timing of mutation to the timing of new vaccine. And we can tell if the virus is changing to escape from our vaccine or not. So this is possible by doing like evolutionary analysis, by comparing the change in the viral genome and proteins, compare with what we know about the environment, about the clinical data, about the vaccine data. We can reconstruct what happened on the evolutionary scale. Okay, so hopefully these three examples will give you enough inspiration that's why we all should think about genomics and other omics data in the perspective of evolution, in the perspective of change, in the perspective of comparative analysis. It's not just a single genome, but we have to compare it to other genomes to understand even more about the gene and protein in our cells. Okay. So just to summarize, we want to start with setting up like the perspective of evolution, because this concept can be used in broad biology, not just people who study evolution. You can identify genes, identify region of the protein to eat it in day-to-day experiment. Okay. Good. So now let's talk about the computational techniques that we need to know in order to be able to analyze the data. So the most fundamental technique is phylogenetics. So phylogenetics is the study of the evolutionary relationship between organisms. Okay, so basically we want to reconstruct this type tree. You might have seen a lot of these before. So this tree tells us which species are closely related, and the branch length tell us how close they are. So if the branch is short, then that means these are very similar. They diverge from each other a short period in the past. But if the branch length are long, this tells us that these species diverged very long ago in the past. So they probably have accurate different mutation along the way. So phylogeny tells us about the time, the timing of how these species diverge from each other and how similar they are from each other. So there are several aspects of phylogeny that we can focus on. We might just want to focus on the grouping of the species. We just want to say that taxon B and C are similar and D is not, D is further away. In this case, we don't even care about the branch length. We just want to see the grouping. We focus on the clustering of the species. But if we also want to focus on the similarity, we want to say that B and C, okay, they are grouped together, but they are not that similar. We have to go back all the way in the past before they diverge from each other. And the timing of divergence here is almost the same as when A diverges from B and C. So the branch length tells us the timing. But sometimes we not only care about the time, we want to see the mutation rate. So the difference between chronogram and phylogram, notice first thing is that for chronogram, all the taxons are aligned because this is the present, the time at the present, 2025. So the time is the same. In order to have the reconstruction of the history where the time is the same, you have to make certain assumptions. which is called a molecular clock. And that assumption is that everything evolves at the same rate. So taxon B, taxon C, like might be human, monkey, mouse, chicken. We assume that they have the same mutation rate. Otherwise, we have no idea how much time has passed. We have to assume that they have the same mutation rate. So if we see more mutation in human, then we assume that there's a lot of time has passed. But if we see few mutation, that means short time has passed. But sometimes we don't want to assume that the mutation rate are the same, right? Because who knows whether human and mouse have the same mutation rate? Probably not the same. So instead of fixing the time, right, to make sure that every taxa are aligned at 20 to 25, we can also reconstruct the phylogeny by only caring about the mutations. Right? How many mutations occur? If a lot of mutation occurs, then you get a long branch. If a small number of mutations occur, then you get a short branch. And we no longer care about that the time has to be 20 to 25 all the way. We want to show that there are more mutation here and less mutation here. So these two can give different information. Chronogram tells us about the timing of the common ancestor when the species diverge from each other. But phylogram, which doesn't care about the time, want to tell us about the amount of mutation that accumulate in each species. So it's two different things, slightly different things. But for now, we are not going to worry too much about these kind of technical details. We want to focus on if we have DNA sequence data, we have genomics data, how can we reconstruct these tree? Right? How can we even get to these phylogenetics tree? So the ingredient that we need three things. First, we have the DNA sequence, we have the protein sequence. We have to be able to align the sequence so that we can tell which species are similar, which species are not. And this is something that we already covered in past lecture. So today we're going to cover the other two topics. First, we need to think about the evolutionary model. How likely it is to see an A change into a T, how likely it is to see a T change into a G. We have to have some models. Otherwise, we cannot reconstruct how much mutation occur. And the last component is how can we identify the tree? There are many trees that are possible. Let's say we have 4 taxa, 4 species. There are many ways to connect these together to form a phylogenetic tree. And these are all different. This tells us different story of evolution. How can we identify the one that fits our data? And I just want to mention, for those of you who might go on to study animal, non-molecular data like phenotype, shape of their physiology, can also serve as a kind of information for reconstructing the similarity. As a molecular people, we look at the genome, look at the protein sequence to compare the similarity. But we also know that similar genomes and similar protein also imply kind of somewhat similar phenotype. So some people also use phenotype, like the color of the feather or the shape of the beak. Or for plant, it might be the shape of the leaves or other aspect of the tree to also drive the reconstruction. Animal with similar phenotype should also be more similar to be compared together with their genomes. So this is also possible. But for this class, for understanding the computational aspect, we're going to focus on just molecular data because we can quantify them better than phenotype. How do we kind of turn the big size into number, big shape into numbers? Okay. So because we know how to do sequence alignment, so we are going to think about the second component, which is the evolutionary model. And essentially, in this case, it's going to boil down to nucleotide or amino acid substitution model. like how likely it is to see an A mutate into a different nucleotide, for example. And we actually have seen this before, but in a different name. We have seen sequence alignment scoring. A match is +1, a mismatch could be -1 or -2. We have seen it before. In this case, we say that if there is no mutation, then it's +1. If there is a mutation, regardless of whether it's A to T, A to G, A to C, they are all -2. So sequence alignment score is a very simple substitution model. It only cares about whether the sequence is the same or different. But this is not how mutation occur in nature. Mutation doesn't occur with the same probability for every pair of nucleotide or every pair of amino acids. So people from way back when you see this number, and you're going to see this number a lot, like 69, 80, 85, these are years, 1969, 1980, 1985. These are the times when people start to have DNA sequence data. This is way before DNA sequencing. This is way before high throughput sequencing. But people start to have like PCR techniques. They start to have developed the Sanger sequencing. So they start to have DNA sequence data. So mathematicians and statisticians start to think about, so what kind of probability model should we use to describe mutation? Back in 1969, the first model, if you even want to call it a model, is that everything is equally likely. So we assume that the frequency of nucleotide in the genome is equal, all one quarter. In the genome, we have 20% being A, 20% being C, 25% being A, 25% being C, 25 being G, 25 being T. And we know that this is not true, right? Because we have different GC content depend on the species. Right? So we know that the assumption that we have equal A to C to G to T is not true. But in terms of a mathematical model, it's a simple one that people can play with, right? Way back when. And then people start to think, because nucleotide, like if you recall back to your biochemistry class, like the structure for the nucleotide can be separated into two groups, right? So people start to model that maybe A and G are more similar and they are more likely to be mutated into each other, and C and T are similar and they are more likely to be mutated into each other. So we start to have two mutation rates. One between AG and between TC, and one across the groups. So the model became slightly more complicated. And then people start to think, wait, we have different AG, ATCG proportion in the genomes. Like in different genes, we have different proportion of ATCG. Can that be a new variable? So people start to incorporate different proportion of the nucleotide. A, C, G, T can be different probability. So we have a slightly more complicated model. So just to summarize, when you see JC, this is from two people's name, Juk Cantor. So they assume that all the nucleotides have the same frequency, 1/4, and all the mutation occur at the same rate. A can become T or C or G with the same probability. The second person, K for Kimura, assume that there is two different mutation rates. One is the transition, one is transversion. So instead of one mutation rate, now we have two. And then three scientists, Hasegawa, Kishino, and Yano, propose that we should consider nucleotide to have different frequency on their own. So we start to have three more parameters to describe the distribution of nucleotide. So if you think about this in terms of how many parameters do this model use? if we write out the Joule-Cantor model where all the mutation occur at the same probability. So the probability of A turning into a C, A turning into a G, A turning into a T, or C turning into an A, C turning into a G, all the off-diagonal are the same. These are the mutation rate. Can change any nucleotide to any nucleotide with the same probability. So there's only one parameter. Whatever mutation rate here is the same for every off-diagonal. And the diagonal have to be 1 minus 3 times this because it's probability, have to sum to 1. So essentially, this entire substitution table only have one parameter. Whatever this x1 is, you can fill in the numbers for the entire table. So we can say that Joule-Cantor model have only one parameter, which is the substitution rate, whatever this x1 is. If we think about the Kimura model, so the Kimura model says that between A and between A and G, there is one mutation rate. But from A to C and T, which is across the group, we have a different mutation rate, x2. And we can fill in everything in this table with just x1 and x2. If it's a nucleotide within the same group, we got x1. If it's a nucleotide across group, then we get x2. And the diagonal, I forgot to put 2 here. It should be 1 minus x1 and then minus 2x2. And so the diagonal, we can fill in with whatever complement the other numbers. So this Kimura model, people also call it a Kimura 2-parameter model because it has 2 parameters. And for the HKY, Hasegawa, Kishida, Yano, they have 5 parameters. So they have 2 mutation rates, same as Kimura, and then 3 more frequencies for A, C, G, and T. So it has 5 parameters. The number of parameters is going to come into play later on. So, but for now, I want you to kind of recognize that for each of these models where we increase the complexity slightly at the time, we add more and more kind of parameters to it. And the model that you're going to see a lot in literature, and you should know, is called a general time reversible model. The name sounds like a physics theory. So the reason why they call this a GTR or general time reversible model is because when we reconstruct the evolutionary history, one assumption that we have to make is the flow of time. Evolution occurs from past to present. But when we have 2 DNA samples, 2 genome samples, how do we know which one occurs first? We actually have no idea. Did we or are different monkeys who kind of emerge first in the evolution? Who knows, right? So if our evolutionary model has the property that is time reversible is a property that if you switch the 2 genome, let's say you assume that human comes before monkey, or if you assume that monkey comes before human, if your model is robust in the sense that whichever choice you pick, you get the same analysis, you get the same result. Right? You're safe. Because even you make the wrong assumption which species comes first, you still get the same tree. So your result doesn't really depend on the choices that you make in terms of the assumption. So people call this time reversible model because it's assumed that the probability that an A will change into a different nucleotide, let's say a G, is going to be the same as the probability of seeing a G turning back into an A. So times going forward is equivalent to going backward. Your substitution metric is symmetric. If you see an A to a G, or if you see a G to an A, this occurs when you kind of flip the order. You get the same probability. So this is a good model. It's a robust model, which is good because we don't know the answer, so we have to make assumptions. So this model has 6 parameters for the substitution rate. Because we select 2 nucleotides, we have 1 substitution rate. A to G equals to G to A. So A and G have only 1 rate. If we pick A and T, the property of A changing into a T or T changing into an A is the same, so we only need one parameter. So there's only 4 nucleotides. If we pick 2 at a time, we can do this 6 times, 6 ways. So we have 6 parameters. And we have 3 different parameters for the frequency of A, C, G, and T. So this model has 9 parameters. In some papers, they might say 8 because they can reparameterize it, but for the purpose, for the sake of being simple, we are going to think of this as 9 parameters. Okay. So that's the idea for nucleotide. Right? For nucleotide, we have no other information, we have no other ideas going on besides the fact that one nucleotide can mutate into another. And it depends if we impose, the substitution rate to be the same throughout, to be the same within group, different across group, or to be symmetric. We can only impose some ideas, but we have no other information about how nucleotide will change. Things became different with amino acids. We mentioned before when we talked about protein sequence alignment that amino acids have side chains. So changing from alanine to a valine might be good because it's still hydrocarbon. But changing from alanine to a tryptophan might be bad because now you have the aromatic ring. So amino acids are not equal. So people have several ideas of how to score change from one amino acid to another. We have talked about these two in the past lecture, like bosoms and PAM. These are used for sequence alignment. It's a score that we use to calculate similarity in the context of sequence alignment. But when people want to model the evolution, the change across evolution for protein, they come up with something slightly different, but the concept is going to be the same. For Dayhoff and JTT especially, the underlying idea is the same as PAM. So they are modeling the change in amino acid over one evolutionary time step, and they extrapolate to longer time step. So if you see the JTT option in any phylogenetics software, you can think of it as similar to PAM applied 250 times. And if you see a model called WAG, developed by Veran and Goldman, this is similar to bosom. So they take protein family that have certain kind of degree of similarity, conservation, and they calculate the probability of seeing a change from one amino acid to the other. And they use those frequencies that occur in nature to populate their score table. So for protein, we cannot do a theoretical model like a nucleotide. We cannot impose the change as parameters because there are gonna be so many parameters. Different nucleotide can change to another nucleotide. Amino acid can change to a different amino acid. There are so many possibilities, and because of the side chain, we cannot even parameterize if changing from alanine to valine, which are kind of hydrocarbon. How is it likely, more likely or less likely if alanine will change into a glycine, which is also hydrocarbon? We have no information to parameterize them. So we need to use the frequency of mutation that occur in nature in order to estimate this score. But because protein have position information, when we talk about protein blast, we talk about side blast, where we know that in a protein domain, the first residues and the second residues can have different properties, different functions. Some residues bind to DNA, so the change in amino acid is going to have more effect than residues that are just part of the structure and not part of the function. So in the context of evolution, people also try to come up with similar idea as side blast. So they want to create the substitution model that have position-specific scoring. So similar to the concept of position-specific substitution matrix that we saw earlier. So for example, they might take a protein family where we know the structure data. So in the past, we have to solve the crystal structure. Nowadays, we might just put all these in AlphaFold. So we got the structure. We can look at the structure and see how they interact in terms of the physical forces. Do they form hydrophobic pocket? Do they have charge interaction? Amino acids that interact physically or via any forces, these positions are going to be evolving together. They are not going to evolve alone. So using information from either the structure or from the functional domain of the protein, some people propose that we should have a substitution model for protein that take this information into account. But again, these are not very popular because it's difficult for you to have the 3D information or have the functional information for every protein that you are going to build the evolutionary history for. So these are not that as popular. But what we should use, if you have protein data, and this is my recommendation, and it's something that you're going to do in your assignment, because for protein, like the trade-off, so let's think about nucleotide and protein. For nucleotide sequence, the trade-off, the good thing is that mutation occur in nucleotide. So we can develop mathematical model that capture the mutation on the nucleotide. But the limitation is that if it's a protein coding gene, right, the change on nucleotide doesn't mean anything on its own. You have to translate the change at nucleotide level to change at protein level before you can understand what phenotype or what function change can occur. So the trade-off for the protein, like if you align the protein, you're going to have the functional information because you are analyzing the nucleotide, the amino acid. Sorry. But the problem is you don't have any mathematical model for the change in the amino acid because that's not how mutation occurs. The mutation doesn't occur on amino acid; it occurs on the nucleotide. Right? So people propose a mix between the two, which is called a codon substitution model. So the idea is every protein must come from RNA, which comes from DNA. So when we have DNA sequence that's coding DNA sequence, like it got translated into protein, we can align the DNA. We want to align the DNA because that's where we can apply our mathematical model. But we want to guide the DNA alignment by the amino acid information, by the codon structure. And we can do this because change at the codon level, like when we have, let's say, TCG codon, like which code for serine, if we have a mutation from C in the middle into an A, that became a stop codon. If we change C in the middle into a G, then we get a tryptophan, so a change in the amino acid. But if we have a mutation on the G, on the third position, like G into a C, we still have serine. G into a T, we still have serine. So we can model the mutation at nucleotide level that each of these mutations can occur on the nucleotide level, they can occur with the same probability. But when we take into account their amino acid consequences, we know that the mutation from C into A is more severe. It became a stop codon. So your protein is going to be truncated. If you change from C to a G, then you get a new amino acid, which can be bad. But if you change a G, the last G, into an A, a C, or a T, it doesn't change amino acid. So these are more likely because this mutation can occur in nature and it doesn't change amino acid. Like these are synonymous substitution. So it's the nature can allow it to exist because it doesn't cause any bad or any adverse effect on the protein. But any mutation that cause a subcodon, like these organisms might not survive, so it's going to be gone from the gene pool. So it's less likely to occur in our data. So to show you some concrete example, if you have protein coding sequence and you align at the DNA level, this will be an alignment that you might get. So the model try to, the alignment try to match all these similar nucleotide together. And you can see that in other position, it's all messed up, right? The codon no longer align together. But if we align at the protein level, using the codon structure as a guide, we can preserve better the structure of the codon as a unit of 3. So this allows us to reconstruct the evolutionary history using mathematical model on the DNA sequence, but guided by the protein information. So this is my recommendation. If you have coding sequence, try to find if the tools that you are using have a mode for codon alignment. And good news is most of the tools have this. So you can select this option. Okay, so now we talked about substitution model, which is like the theory that we apply to the mutation. Next, we're going to focus on how to figure out which phylogenetic tree, like what shape it should be, which species to group. How do we do that computationally? And first, we're going to review these techniques called hierarchical clustering or agglomerative clustering. And this animation kind of shows you that, let's say we start with the loop cycle. So we start with 7 kind of species, like 7 genomes. The idea is we want to group genomes or species that are more similar together. The red ones, these got grouped first because they are similar. And then the green one, the red one are very similar. And then the green are next. And then the blue is last because these are further apart. So the concept of hierarchical or agglomerative clustering is we're going to do this kind of clustering step by step by grouping taxa or genomes that are more similar first, and then take the one that are further away into the group until we connect everything. So everything at the end is going to be just one cluster that connects everything together. So this kind of tree looks similar to phylogenetic tree, right? Because the algorithm is the same. For phylogenetics, we're going to also calculate the similarity between the genomes and use those similarity score to drive which species to group first, which to group later. So there are several ways to think about this. So there is a very simple technique called UPGMA, unweighted something, I also forgot, but it's a hierarchical clustering procedure. So we calculate the evolutionary distance, which is based on the substitution model between our taxa and any species or genomes that are more similar got grouped. So this process is essentially the same as the animation that you saw before. It just have different names because people in different fields come up with similar techniques, right? And they name it differently. But the concept is the same. We're going to group species or taxa kind of one at a time until we connect everything together. Another approach called neighbor joining. The idea is we're going to start with what we call a star tree. This is called a star tree. So we connect every genome together in the middle. So basically, we have no idea who diverged first. So we just say that, okay, everyone comes from the same common ancestor. But we don't know yet who diverged first, who diverged last. So everything became dashed line. Dashed line means we haven't decided yet. But we know that if we go back far enough, everything comes from a single common ancestor. So the common ancestor must be here. but we are not sure yet how to connect the species that we see today. So the idea is we want to group them together one at a time. So the set looks similar to hierarchical clustering, but the visualization is different. This visualization is based on convention of people in evolution. So we start with a star graph, and let's say we find that F and G are taxa that are most similar. So we're going to connect F to G and create a new branch here. So the earlier part is still a dashed line because we don't know yet how the common ancestor between F and G will connect to the global common ancestor. But we know that, okay, F and G should diverge last. So we connect them first. And we call this common ancestor U. So next we might find that, oh, E is very similar. So we connect E to this group. So we connect E upstream of U, and we get a new common ancestor, V. And we keep doing this until all the branch became connected. So it's similar to hierarchical clustering, just visualized differently. And it's called neighbor joining because we are joining the neighbor together. We are joining the neighbor together. We are joining the neighbor together. So the name kind of is very intuitive. And the way that they calculate kind of which neighbors to join. So first, they calculate two things, and I color code them so that we can understand the motivation better. So first, if they want to join taxa I and J, like two genomes, first they calculate the similarity between I and J. Are they similar or not? And then they ask if I and J are I and J similar to other genomes, the rest of the genomes, are they similar to I and J? So basically, if we want to group I and J together, it's a competition between two considerations. First, I and J has to be similar, so that we want to connect them together. But at the same time, we don't want other genomes to be similar to I and J, because otherwise we would connect I and J to those things instead. Let's say I and J are similar, but you can find a new genome K that is even more similar to I than J. Then you will connect I to K instead. So this is a competition between similarity within I and J versus similarity between I or J with other things. So the orange is a penalty. The green part is the score. We want to subtract these two, and if I and J are similar to each other, but not similar to others, then we want to connect them. So this is the criteria for neighbor joining. And if you're wondering why do we have kind of n-2 here, it's because the orange term here has I and J like fixed, and then the other genomes, there are n-2 of them. So the summation here have n-2 terms. So these green terms also have to multiply by n-2 so that the scales are the same. Okay. Another technique for grouping DNA sequence together, grouping genomes together, is called a maximum parsimony or minimum evolution model. And this is a kind of Occam's razor principle. So Occam's razor idea is that if you have two possibilities, one is simple, one is very complicated, we prefer the simple one. In the context of evolution, so let's say you have two possible explanations for the evolution. Like one requires 1,000,000 mutations, one requires 10 mutations. So we would think that during the course of evolution, if 10 mutations is enough to create a new species, nature shouldn't make 1,000,000 mutations. So we prefer the simple one. So in this case, if we want to reconstruct the phylogenetics from these genotypes, we have two possibilities. One of them has only one mutation. So the common ancestor is a G, and there is one G to T mutation here. And it creates two resulting species. So only one mutation can explain the genotype. Versus the second explanation, assume that everything is a G, like the common ancestor is a G, and then somehow species 3 and 4 both have the same mutation, G to T, which are not related to each other. Like species 4 diverge first, acquire the new mutation. Species 3 diverge later, and then acquire the same mutation. So this can occur. There is a concept called convergent evolution, where two unrelated genomes acquire the same mutation without being related to each other. This can occur because you can have two organisms live in, let's say, different continents, like one in Australia, one in Europe, but they face the same environment, they face the same condition, so they acquire the same mutation, and this can occur. So, but it's unlikely most of the time. If you see the same mutation in species that can be related, usually they occur. This mutation can occur once, sometimes in the past. So that's why maximum parsimony and minimum evolution is a simple way that we can use to get a quick estimate of the phylogenetics for our data. One situation, so we talked about convergent evolution where this assumption can fail. Another situation where this assumption will fail is when you have a long evolution. If we try to compare human to dinosaur, if we go back that long in the past, right, it's possible that a mutation can occur and then it's reverted. Because so much time has passed that an A might got mutated into a T, and then at a certain point it got mutated back into an A. Right? Because enough time passed, you can have multiple mutations on the same position, which can revert back any changes. So in this case, the assumption that the smallest number of mutations is the right one is no longer true. There is no mutation that we can see, but actually there are two mutations underneath. So this violates the assumption. Okay. The technique that we want to focus on, which is the technique that people use most to reconstruct phylogenetic tree, is called the maximum likelihood principle. This is something that you have seen in statistics class before, but probably not with this term. So let's talk about it a little bit. Okay. So I think from two lectures ago when we talked about hidden Markov model, like we have some use of probability terminology. So I'm going to take this chance to review that a little and then use that for the maximum likelihood idea. We need this for the maximum likelihood idea. So when we have a certain event, like certain genome, certain mutation, when we say probability that that event will occur, the notation will be P, and then in parentheses, the symbol for that condition, for that event. Joint probability is when we talk about more than one event, like more than one mutation. The probability that SNPs A and SNPs B will occur together. So joint probability, two or more events separated by a comma. So this means the probability that both events occur. The third notation we already saw before is with the vertical bar. So this means conditional probability. The probability that A happens given that B already happened. So if we want to think about this, so let's say A is one variant, like one mutation, B is another mutation. So this means that if we talk about probability of A, like that would be the probability that that mutation occur in the population. If we talk about probability of A and B as a joint probability, then this is a probability that you will find an individual with both mutations, like both variants, together. But if you say A given B, this means your population pool are going to be just people with mutation B. So your population became a new set, a smaller one, where everyone in that pool has mutation B. And you are asking, within that pool, what fraction or what probability also have mutation A? You're already given that B occur. So it's different from joint probability, where joint probability, the pool is the entire population, and then you count people with both mutations. For conditional, your pool is people with only B mutation. So it's smaller. And among that group, within that group, you are looking at people with also with mutation A. So the mathematical relationship, conditional probability, is equal to joint probability divided by the probability of event B. So one identity that you are going to have from this relationship between joint probability and conditional probability is that you can write joint probability in two ways. One, you take B as the condition. The other way, you take A as the condition. Both multiply them together, get the joint probability. So now you have this identity that we are going to use later on. Another thing that we want to talk about is we see here that we have probability of A kind of condition on B, and then probability of B condition on A. Right? As a mathematical symbol, it doesn't make much difference to you, like whether A or B is given. But I want to show that when we think about data or even in biology or in nature, anything in nature, there is an order that one makes more sense than the other. Right? So let's think about the example that we have for the hidden Markov model. We think about the weather, like sunny or rainy, and we think about a person's activity, whether he or she goes shopping or walking or cleaning his or her apartment. The weather comes first, and then the activity of the person follows. Whether we go shopping or staying at home, we look at the weather first. So there is a natural order of things. So if we say property of shopping, whether we go shopping, given that the outside is sunny, make some sense, make more sense. Then if I say, what is the probability that the outside is sunny, given that you are shopping today? Right? Whether you are shopping or not doesn't make the sun comes up or down or anywhere, right? So in terms of biology or nature, certain order make more sense. Okay. And we notice that this one makes more sense than the other. So let's keep this idea in mind. We're going to think about A and B in terms of phylogenetics. So let's say A, and I'm color A in black and B in blue. So A is an event where you observe some evolutionary data, like the genomes, for example. You observe the genomes. And B is the event that the phylogenetic, the tree that you are producing, that the tree that you get is true. Which one makes more sense? Probability of A given B or B given A? So let's think about this. Right? Let's start with A given B. So this means if I assume that the phylogenetic is correct, what would be the probability that I will see these genomes? I would say that this makes more sense because if you know the phylogenetic, like you know the tree, you can imagine that if the nature follows this tree, it will produce the genomes according to the divergence in the tree. But instead, if you ask, what's the probability of B given A? The probability that the tree is correct, given that this data is observed. This can also make sense because from our perspective, which one do you see first, the data or the tree? You see the data first, right? You get the data and you build the tree. So one of them makes sense in terms of the sequence in nature. One of them makes more sense in the sequence based on your experience. So in this case, the purpose of the statistics is probability of B given A because we saw the data and then we want to prove that the model is correct, the tree is correct. So this is the probability that we want from our data. But the problem is, the probability of the tree is true given the data is the one that we want in terms of statistics, in terms of human perception. But it's difficult to calculate. If you have the data and you have so many possible phylogenetic trees, how would you quantify the probability that your tree is correct? But if I ask you, like, if I give you a tree, and I ask you, how does the data fit the tree? This is simpler, because you already have the tree being fixed, and we are asking, does the data that we are producing, does it match the tree or not? So if we go back to the maximum parsimony example, so I give you these two trees, and I ask which one match the data. You can tell at a glance that this one looks better. So if I give you the tree, and I ask you if the data match it, it's an easy problem. But if I give you the data, and I ask you which tree is correct among our other trees, it's difficult because there are more than 2 trees. There are also millions of other trees. So now we have a situation where one thing is easy to compute, A given B, this is easy to compute, but it's not what we want. We want B given A. So Bayes' rule and this relationship between the conditional probability comes to play because we can write the thing that we want as the thing that is easy to compute. So we change probability of B given A using this formula to get the probability of A given B, which is easier to compute, times the probability of B divided by probability of A. And I'm getting out the probability of A because the probability of A is the probability that you will observe the data. It's a constant. The probability that you will see this genome is a constant. You're going to stumble into this genome data. There's no chance to it. The term that is important here is the probability of B, the probability that your phylogenetic is true. And if we know nothing, if we know nothing about this kind of species, about the evolution, we can assume that P probability of that this thing is true is just a constant, and we can ignore it. So what I want to say is that we start with what we want, B given A. We use the Bayes rule to transform it into a term with into an expression with 3 terms, A given B, B, and A. We can cross A out because that's a constant. In most cases, we can also cross B out because we have no idea. So what's left is we can estimate what we want with what we can compute, which is A given B. So this probability of A given B, where A is the data and B is the model, the special term for it in statistics is called likelihood. The probability of observing a certain data given a certain model or given a certain hypothesis, this is defined as likelihood. So the likelihood that you will observe the data given that it's produced by a certain model. So the idea of maximum likelihood is that we want to change the model because we have options like on the model. We want to change it until we can maximize the likelihood. So we want to optimize this function, this probability of data given model, by changing the model. The data doesn't change. The genome is what you observed. But you want to change your phylogenetics. You want to change your model until you can maximize this kind of expression. And what happens when you maximize this expression is you are maximizing what you want as well. If you maximize probability of A given B, given that the other are constant, you are maximizing probability of B given A. So maximum likelihood is a shortcut to getting this probability to be maximized by doing only thing that we can do, doing all the things that we can do. So this became an optimization problem again. So that's why when I think in the first lecture, when I mentioned that one of the key kind of concepts in computational thinking is optimization. In this case, we want to optimize the model, the phylogenetic tree, in such a way that the likelihood of our data is maximized. And we can do this in several ways, whether with calculus or with brute force, like we can change the model over and over and calculate the likelihood and then pick the one that's higher. So let's try some example, and we will go over this very quickly because I trust that you can do them. So let's say we have gene A with 2 alleles, capital A and small a. In a study of 1,000 Thai individuals, we found 700 with genotype capital A, 200 being heterozygous, and 100 being small a. If I ask you what is the allele frequency for the capital A, you can do it by just, okay, we have 700 individuals with 2 capital A, right? So we have, and then we have 200 heterozygous. So in total, we have 1600. We have 1600 alleles of capital A. And we can do the same for the small a. We can count from our sample. So your frequency is going to be 0.8. Let's say another example. Okay, let's skip this one because I think the amount of time. So let's stick with this example where we have a certain genotype and we want to estimate the allele frequency. You can do this and you get 0.8. You may ask yourself, am I following any principle when I calculate this as 0.8? And you might be surprised that you are actually getting the maximum likelihood estimate. So let's see how we derive 0.8 by applying the maximum likelihood principle. So we write the frequency of A, let's say as a parameter, as a small p. Okay, so let's try to reproduce your answer like that we get with no thinking at all, like 0.8, with the maximum likelihood principle. So we parameterize, so we define parameters, small p, to be the frequency of the capital A allele. So for small a, it's going to be 1 minus p. So what happens is, if this is the allele frequency, like we know the probability of selecting a Thai person and get to capital A would be p squared. To get heterozygous would be 2 times p times 1 minus p. And then this 1 minus p squared. So we know this from genetics. So if we write out the likelihood, what is the probability that we see 700 people with genotype capital A, 200 heterozygous, and 100 small a? We can write it. It's going to be the probability of seeing capital A times itself 700 times. And then the probability of heterozygous to the power of 200. And then the small a to the power of 100. So if we substitute all these in, like we get something very scary. But at the end, it's just 2 and p and 1 minus p. And to the a lot of power. If we want to identify p that maximize this, right, going back to your calculus, we can just differentiate it, set it to 0, and you will be surprised to see that you get 0.8 again. So when you estimate it by counting out the alleles, you are actually following, you're getting the maximum likelihood estimate, but without knowing it. In the other example that I skipped, I'm going to mention just quickly, like if we take some cancer patient and their survival data, and we estimate what's the annual survival rate, you're going to get something 18/23 by just kind of counting out how many years that the patient survived, how many years that they died, basically. 18/23. If we follow the same thing with maximum likelihood principle, set the survival rate to be R, and then again calculate the likelihood out, you get something with a lot of power. But at the end, if you differentiate it, set it to 0, you get back 18/23. So many things that we estimate by hand coincide with the maximum likelihood estimate. So it's a powerful thing that we do without knowing. Okay, so that's a fun kind of connection. Okay, so let's see how we apply this to phylogenetics. So for phylogenetics, the likelihood is the probability that we see the sequence data, the molecular data, the phenotype data, given two things. That the substitution model is correct, like the one that we picked, and the phylogenetic tree that we are going to produce. So we have two components in the model, the substitution and the phylogenetics. Good thing is, we have only a few numbers of substitution model. Good thing the statistician don't kind of create thousands of models for us. But the problem is, how many phylogenetic trees do we have? If we have 10 species, do we have a lot of possible phylogenetic trees, or is it not that many? So let's try to think about this, and don't worry too much if it's too complex. But I just want to kind of show you how many trees can be produced based on just a few kind of numbers. So let's say we have this kind of unrooted tree. So unrooted means we didn't specify the common ancestor. Like we don't know where the common ancestor occur, whether it's here or here or here or here. We don't know. So this unrooted tree, so it's a tree. Maybe I should have introduced graph theory, but anyway, for now, let's say this is a tree. And if we have n leaves, so leaves are the endpoint. In this case, we have 1, 2, 3, 4, 5 endpoints. We can count. If we have 5 endpoints, we're going to have in total 8 nodes, counting the internal one. If we want to add more species, more taxa, to the tree, what we are doing is we are picking a certain position in the tree, and we add a new branch. We are doing this every time. So we add 2 nodes at a time. So basically in this graph, in this tree, we can calculate if we have 5 leaves, 5 species, we have 8 internal nodes, and we have 7 branches. You can count. We have 7 branches. And I'm writing this out here as you can replace this with any n, but we can look at the numbers on this small graph just to understand. And the number of branch also goes by twice every time we add a new node. So that's why we have 2 times n. N is the number of leaves. So for each unrooted tree here, when we want to add a new node, we create 2 more nodes, we create 2 more branches, and we can do this at many positions. We can add between 7 and 8. We can add this between 4 and 8. We can add this between 5 and 8. We can add this between 1 and 7. So we have many choices to add the new genome, to add the new taxon. So from each tree like this, we have this many locations to add the new leaf, many places. So if we calculate all this out, the number of trees where you haven't put the ancestor yet, if you have n taxa, n genome, you're going to have this many possibilities. So if you recall factorial, like n factorial is n times n minus 1, n minus 2 times n minus 3, all the way to 1. That's already large. In this case, it's going to be more closer to 2n factorial. So this is the number of trees. If you want to put the ancestor, if you have 10 taxa, you have 34 million possibilities. So this is why for optimizing the substitution model, it's still possible to try out all the models. And in the assignment, you're going to try out many models. But for the phylogenetic tree, it's impossible to try out all the models. So we need a technique called heuristic. So we mentioned quickly in the first lecture that when we cannot optimize perfectly, we have to do some guessing. And we can do that with what we call heuristic algorithms. So in this case, we need heuristics for searching the phylogenetic tree. How do we identify the tree topology among 35 million, the one that looks good? So people propose many techniques, and you will see these in the software that you are using for the assignment. So that's why I show them here. So for example, NNI, this is a nearest neighbor interchange. So they're going to pick a certain branch in the tree, and they're going to flip the neighbors on the 2 branches. Notice how this red branch used to connect A, B, C, D. Now it becomes A, B, C, E. So E got switched from the right side to the left side. So if we keep doing this, right, we can generate more trees by changing the current one a little by little. So with this technique, we can sample many trees among 35 million until we are satisfied. We might have to do this 10,000 times until we are satisfied. Okay, we are good. We are happy enough. The best tree that we found among these 10,000 is going to be the one we pick. We have no hope of going to all the 35 million. So other techniques are similar. They're going to take some modification to the tree. So in this case, it's going to cut certain branch and then put it back together. Imagine making a bonsai tree. So you are cutting certain branch and then you are putting it elsewhere. So you generate new tree by editing, by changing the current tree. And there is nothing sophisticated about this. We are just gonna try to make more tree and more tree and more tree, and hopefully not the same tree until we are satisfied that we have sampled enough trees. No better way to do it. Okay. But at least now we know why it's difficult, and you will also understand why when you analyze the data in your assignment, why it will take more than 30 minutes to do so, because it's running these kind of things underneath. But it's what we have to do if we want to analyze real data. Okay, so we have seen the two key components, like the substitution model and the phylogenetic tree kind of algorithms. There are a few more other concepts that I want you to be familiar about when building phylogenetic tree. The first one I mentioned quickly is called molecular clock. So this is when you want the tree to have the time being aligned. So every taxa, you want the year 2025 to be at the same X location, X coordinate. You have to assume that there is a clock. Each mutation occurs at the same rate in every species. With that assumption, we can model the time to be proportional to the number of mutations. But without molecular clock, we're going to get trees similar to the bottom rows here, where none of the taxa will align. So you cannot make any inference about the time, but you can still see the change, the mutation that occur on each branch, if it's long or short. So these can have different purposes. The second one is some nucleotide position or amino acid position are constant. So this doesn't change over time. If you apply the same substitution rate on position that never change, if you're going to have a very skewed score, right? Similar to how we talked about GC island before. If you apply the same substitution model or the same scoring criteria to position on your sequence that have different evolutionary model, you're going to get very skewed score. So some software allow you to model some residual position or some nucleotide position as what they call invariant. So invariant means that this position rarely change or doesn't change at all. It will be modeled with a different probability. And even among position with mutation, we know that mutation at different locations on the genome probably are not going to follow the same probability. So in several models, they allow you to say that, okay, for position with mutation, there might be different mode of mutation. So different substitution rate, basically. And some model will say that there will be 5 mode of mutation. Some models say there will be 8 mode of mutation. In real life, people will try to adjust it up to a certain point. But for the sake of this class, we are not going to worry too much about like how many mode of mutation should there be. But we should be aware that some models allow the substitution rate to change from one position to the other. And the keyword, they will call this a site-specific mutation, site-specific. And also, sometimes they will call this a gamma-distributed mutation rate. Because when they draw the mutation rate for each site, they draw it from a gamma distribution. And for those of you who hasn't seen a gamma distribution before, it sounds scary, but the shape is very simple. So gamma distribution, depending on the shape parameter, it can behave like an exponential decay, like the blue line here, or it can behave like a Gaussian distribution, the cyan line here, or it can behave like a skewed Gaussian, like the red line here and the green line here. So the gamma distribution is used a lot. This is the first time that we are seeing it here, but it's not going to be the last time. The gamma distribution is famous because it's versatile. You can parameterize gamma distribution to be exponential, or to be Gaussian, or to be skewed. So you can make it mimic many things. So people use it for many statistical models where we don't know what goes on. So it's like a flexible case. We talked about non-synonymous and synonymous substitution before, and that's the concept of natural selection. But I want you to see the symbol. If you see any symbol that says dn and ds, so dn would be the non-synonymous substitution rate, or the rate when the substitution changes the amino acid. If you see the ds, it will be the synonymous substitution rate, or the rate of mutation where the amino acid doesn't change. And people like to calculate the ratio between these two, like dn over ds, because like if mutation occurs, like sometimes it will be synonymous, sometimes it will be non-synonymous. If the protein is being conserved, right, that means when there is a non-synonymous substitution, it's bad. If you observe the data, most of the mutation will be synonymous, because non-synonymous mutation occur in some individual is bad, so that individual got kicked out of the gene pool. They don't pass on their mutation. So if you see DNDS being low, that means the protein is being conserved, because DNDS being low means DS is high. DN is low. But if you see DNDS being high, like much greater than 1, that means the protein is evolving. It's accumulating new mutation that change the amino acid. So in bacteria, usually, you will see that if you treat it with drugs, and the protein is evolving to survive against the drug, DNDS is going to be high, because the bacteria is trying to evolve so that it can fight with whatever compound that you are giving. Okay. But for essential protein, the DNDS is going to be small, because for essential gene, the nature don't want to change, organism doesn't like the change to the amino acid sequence. Okay, so I'm going to go over time a little bit. Okay. So we mentioned before that we have multiple nucleotide substitution model. How do we know which one is good? So in statistics, there is a concept called nested model testing. The idea of nested model is when you have multiple models that are increasing in complexity. So you have the Juke Cantor being the most simple, and you have more complex models that have more and more parameters. The idea in statistics is, if the model is more complex, it's usually bad, because that means you are increasing the degree of freedom. But the good thing is, if your degree of freedom, the parameters that you add, are the ones that actually needed to explain the data, then you get better likelihood. So nested model testing is a competition between the positive score, which is the likelihood, like more likelihood the better, versus the negative part, which is the number of parameters. The more parameters, the worse. If we have two models that have similar likelihood, one have more parameter, one have less, we're going to pick the one with less parameter. Because if both of them explain the data equally well, we want the less complex one. So when we run phylogenetic analysis result, and we try multiple substitution model, we're going to get different likelihood. With more complex model, getting better likelihood. But because more complex model have more degree of freedom, we can do chi-square test to compare the change in likelihood versus the change in degree of freedom, and see if the p-value is significant. If it's significant, then we accept the we reject the simple model and we take the complex one. If the p-value is not significant, then we reject the complex model and we stay with the simple one. In the assignment, you see some table like this, and you have to make interpretation which model is the best. But now we know like the principle, so you can apply. So we look at the likelihood, and then we look at the number parameters, and if we want to do statistics, we use the chi-square to calculate the significance. Another keyword that comes with phylogenetic analysis is called bootstrapping. So bootstrapping is the concept in statistics where we want to sample the data multiple times so that we get an estimate for the variance. Imagine like we have one genome for each species. If we build a phylogenetic tree, you're going to get only one tree because we will have one data point for each. But at the same time, we want to know if our tree is trustworthy. Do we have confidence about it? So we want to be able to build multiple trees. And if multiple trees agree, we are confident about the tree. If multiple trees are all different, like they group species differently, then we have a problem. Our data doesn't clearly determine how the species evolve. So how can we start from one data set and create multiple trees? So imagine that if we study a genome, let's say for bacteria, with 4 million nucleotides. Basically, we have 4 million positions to calculate the tree. Each position has its own genotype. So instead of using all 4 million genomic positions to build a tree, we can divide or we can sample 1 million positions at a time, drawn from the 4 million positions. Each time we build one tree from 1 million positions. Each time we build a new tree from a new 1 million position. So we can make this, let's say 100 times. So we make 100 trees. And we will have this estimate called bootstrap support that says that among 100 trees that you make, all of them group these two species together. So we are confident that these two are related. If we see 96, this means 96% of the time, these species are grouped together. So we are also confident that these are correct. But if you see like 67%, 15%, then that means that depending on how you select the data, you can have the species grouped together or not. So in this case, you might not be so confident about the tree that you have. And this is something that happens regularly because different genes on the genome can have different evolution. Conserved gene will have a certain evolutionary pattern, and other species-specific gene will have a different pattern of evolution. So people sometimes want to see this difference. They want to know if housekeeping gene evolve in the same way as other genes, like to understand if there's any evolutionary effect on different gene families. So the discrepancy here can be a feature and not just problem. Okay. So the next part is also included in your assignment. So I'm going to go over quickly because I also add some explanation in your assignment as well. But basically in your assignment, you're going to analyze phylogenetics using a software called Mega. This is not a best tool, but it's the easiest for you to use. It has a graphical interface. It runs on Windows and Macintosh. So in this tool, you're going to build the phylogenetic tree, and you're going to see a lot of these options. Again, don't worry, this will be explained again in your assignment. But you're going to see many options. But most of these options you have seen before in the slides earlier, like the substitution model. The name, this name you have seen before, so you know kind of what they are. You have seen the meaning behind bootstrapping before. You have seen the term site-specific evolutionary rate before. And you have seen all these tree building, like nearest neighbor interchange, and other things before. So hopefully you'll be able to make sense of how to run the phylogenetic analysis through the tool. And to show you that all the terms that we see in the lecture are the terms that you encounter when you analyze actual data. So the output that you get from the tree, let's talk about the format a little bit to close out this section. You get the visual, like the tree is a figure. But if you want to save it, to copy it, to edit it, the format is called naming format. And it's just parentheses. So in this case, this parenthesis means that you have a raccoon grouped with a bear. So this is a genome for animal. And the branch length over raccoon is 19.19. And the branch length over the bear is 6.8. And these are then connected together. And then you have a bootstrap support of 50. So 50% of the tree, the raccoon and the bear are grouped together. 50% of the time, they are not. And once the raccoon and the bear are grouped together, the branch length above them, the length is 0.846. So the way that you read, you just read the parentheses from the innermost parentheses all the way to the outermost. So this is a format where you can copy this and paste onto many online tools, and they will draw the tree for you.